Welcome to our deep dive. We're going to be cracking open two essential texts in organic chemistry.
Oh, it's going to be fun.
Essentials of Organic Chemistry and March's Advanced Organic Chemistry.
Two classics.
And we're going to try and grasp the foundations of organic chemistry.
Yeah.
And the thing that I find really fascinating is how we can go from simple molecules like hydrocarbons
Right.
and build incredibly complex structures like DNA.
Really is amazing.
It is. It's like using the same building blocks to make a tiny house.
Yeah.
Or a giant skyscraper.
It's all about how you put those pieces together.
Exactly. So, to make sure we're on the same page,
Yeah.
what are those fundamental building blocks?
Okay.
And what's the glue that holds everything together?
Well, you can think of carbon and hydrogen as your primary materials.
Okay.
When they combine, they form hydrocarbons.
Right.
Now, hydrocarbons can be saturated.
Like the ones we find in natural baths.
Exactly. Those are pretty stable because they only have single bonds.
Mm-hmm.
But then you have unsaturated hydrocarbons.
Like in vegetable oils.
Right. They contain double or triple bonds.
Those pi bonds.
Yeah, those pi bonds are
So they're unsaturated because they have the potential to add more atoms.
Exactly. It's like they have extra slots available for bonding.
Okay. So those pi bonds,
Yeah.
they make the molecule more reactive.
They do. They're formed by overlapping p orbitals
Right.
and create these sites where other atoms can attach.
And that changes the molecule's properties.
It sure does.
Now, what about ethylene and acetylene?
Great examples.
They're both hydrocarbons, but they have different structures.
They do. Ethylene, with its double bond, has sp2 hybridized carbons.
So that gives it that flat, planar structure.
Exactly.
And then acetylene with the triple bond
That one's linear.
because of the spat hybridized carbons.
You got it.
Okay, so hybridization really shapes these molecules.
It's fundamental.
But we've got covalent bonds holding the atoms together within the molecule.
Right.
But are there any other forces between molecules?
Oh, absolutely. Hydrogen bonding is a big one.
Especially in biological systems.
Oh, it's crucial there.
Okay, remind me, how does hydrogen bonding work?
So it's a weak attraction
Okay.
between a hydrogen atom that's bonded to an electronegative atom.
Like oxygen or nitrogen.
Exactly.
Okay.
And then another electronegative atom in a different molecule.
So it's not as strong as a covalent bond,
Right.
but it's still really important.
Incredibly important.
Now, Essentials uses acetylacetone as an example.
Yeah. In its enol form.
What's special about the enol form?
It exhibits intramolecular hydrogen bonding.
Meaning within the molecule itself,
Exactly. And that influences its properties.
like boiling point or stability.
You got it.
Now, carboxylic acids,
Yeah.
like acetic acid,
Right.
those have intermolecular hydrogen bonding.
They do. That's between different molecules
Wow.
forming these attractions.
Now, you mentioned biological systems.
Mhm.
Where does hydrogen bonding come in there?
Well, think about DNA.
The molecule that carries our genetic information.
Exactly. The two strands of DNA are held together by hydrogen bonds.
Between those base pairs.
Exactly. Adenine always pairs with thymine.
Guanine with cytosine.
Right. And it's all thanks to hydrogen bonding.
Wow. So these weak attractions are holding the code of life together.
It's pretty incredible, isn't it?
It is. It seems like these tiny structural differences can have these massive consequences.
They really can.
Now, I'm looking at these 2D representations of molecules in the book.
Oh, yeah, the Fisher projections.
Yeah, but how do they actually translate into 3D?
That's a great question. Fisher projections are useful for sugars,
Yeah.
but they have their limitations.
So they don't tell the whole story.
They don't. To truly understand a molecule, we need to think 3D.
Especially those staggered conformations.
Right. That's where the energy is minimized.
Okay, so we got to think about how these molecules exist in 3 dimensions.
It's crucial.
I'm also seeing these fused ring systems.
Oh, yeah. Like decalins and hydrogenes.
So 2 rings sharing a common bond.
Exactly. And they can exist in cis and trans forms.
So it's like 2 hoops linked together.
I like that analogy.
But they can be linked in different ways.
Right. And their stability depends on how they're fused.
So usually the transform is more stable.
Yeah, where the groups are on opposite sides.
But are there exceptions?
Oh, there are always exceptions in organic chemistry.
Of course.
Hydrogenes, for example.
The ones with the 5-membered ring.
Yeah, fused to a 6-membered one.
Right.
They actually favor the cis form.
Why is that?
Well, that cyclopentane ring,
Uh-huh.
it's planar,
Okay.
and that causes strain.
Ah.
So it's better accommodated in the cis fusion.
Where things are more bunched up.
Precisely.
So even though they're both fused ring systems,
Mm-hmm.
those subtle differences
Yeah.
lead to opposite stability trends.
They do.
It makes me wonder,
Yeah.
how does stability impact reactivity?
Good question.
Yeah.
They're often inversely related. The more stable a molecule,
Uh-huh.
the less likely it is to react.
So a really stable molecule is kind of boring.
In a way, yes.
Now, Essentials mentions electron donating and electron withdrawing groups and how they can influence the acidity of phenols.
Right.
So tweaking the structure slightly
Yeah.
can change how easily a molecule loses a proton.
Exactly. It can become more or less acidic.
Now, speaking of acidity,
Yeah.
let's revisit pH.
Oh, yeah. The measure of how acidic or basic a solution is.
Right. And Essentials provides a handy reminder of how to calculate pH.
Using acetic acid as an example.
The acid in vinegar.
Exactly.
And acetic acid is a weak acid.
Right, because it doesn't completely ionize in solution.
But understanding pH is important.
Oh, it's essential in so many fields.
Now, we started with simple hydrocarbons,
Mm-hmm.
but organic chemistry goes way beyond that.
Oh, yeah.
How do we get to those complex carbohydrates?
The ones that fuel our bodies.
Exactly.
Well, it all starts with simple sugars like glucose. And when you link glucose units together with those alpha-1,4 acetyl linkages,
Yeah,
you get amylose.
one of the components of starch.
Exactly. It's like forming a chain, link by link.
Okay.
But it's not just linear chains.
No?
Nope. Amylopectin, another starch component,
Mm-hmm.
has alpha-1,6 linkages.
And those create branches.
Right. It becomes more complex, like a tree.
So it's like a branched chain of glucose units.
Exactly. And our own energy storage molecule.
Glycogen.
Yes. Glycogen takes branching to a whole new level,
So it's like a super branched tree.
packed with glucose for efficient energy storage.
It's incredible.
It is.
But there's more to organic chemistry than just building big molecules.
Right.
What about those reactive species?
You're talking about radicals.
The ones that can cause damage.
Exactly.
Yeah.
They're molecules with unpaired electrons.
So they're highly reactive,
They are. And peroxides are common sources.
like dibenzoyl peroxide.
Yeah, that was a classic.
Now, Essentials brought up vitamin E.
Ah, yes. Alpha tocopherol.
Right. And it's a powerful antioxidant.
It is. It combats those damaging radicals.
So how does that work?
Vitamin E interrupts the chain of reactions.
The ones that radicals initiate.
Exactly.
Yeah.
It donates a hydrogen atom to a radical
So it stabilizes the radical?
and prevents further damage.
It's like a self-sacrifice
In a way.
to protect other molecules.
It's a hero.
Now, this makes me think,
Yeah.
if antioxidants are so important,
Mm.
should we all be taking vitamin E supplements?
That's a great question, but it's best to talk to a doctor about that.
Right, of course.
While antioxidants are beneficial,
Yeah.
overdoing it with supplements
Mhm.
can have unintended consequences.
Ah,
Getting a balanced intake from a healthy diet is a good approach.
so everything in moderation.
Exactly.
Now, Essentials also clarifies how tautomers,
Those chameleon molecules.
right, how they're different from resonance structures.
Ah, yes. Tautomers involve actual atom movement.
Usually protons.
Often, yeah.
And that results in distinct isomers.
Exactly. They can exist in equilibrium.
So they're kind of switching back and forth.
In a way, yes.
Now, the keto form is often favored,
It is, yeah.
but there are exceptions.
Always exceptions.
Of course.
The book uses those 1,2,3 decarbonyl compounds as examples.
Like ethyl acetoacetate,
Right. In those cases, the enol form gets a stability boost.
because of conjugation and hydrogen bonding.
You got it.
So those shifts between keto and enol forms
Mm-hmm.
can impact how these molecules react.
You can.
Essentials also describes the aldol condensation,
Class.
a way to build bigger molecules,
Exactly. By combining two carbonyl compounds.
like an aldehyde and a ketone,
Right. The book uses acetone and benzaldehyde as an example.
and they react to form benzalacetone.
An unsaturated ketone.
So it's like snapping Lego pieces together.
To create something new.
Exactly.
Yeah.
But we can also change existing molecules, right?
We can.
Like D-glucose.
Yes. The book discusses base-catalyzed enolization.
And what happens there?
Well, D-glucose undergoes some surprising transformations.
Like what?
Well, you remove the alpha hydrogen,
Mm-hmm.
and you get an adidyl intermediate.
Okay.
And from there,
Yeah.
it can revert back to D-glucose,
Uh-huh.
but it can also become D-mannose.
Through epimerization at C2.
Exactly. Or even transform into D-fructose.
Wow. So one simple sugar
Mm-hmm.
can become 3 different sugars.
It's pretty amazing, isn't it?
It is. It's like molecular magic.
It is, in a way.
Now let's move on to proteins.
Yes. The building blocks of life.
They're made up of amino acids linked together in a specific sequence.
Right, that's the primary structure.
But what comes after that linear chain?
That's where secondary structure comes in.
Like those alpha helices and beta-pleated sheets.
Exactly. And guess what plays a key role there?
Hydrogen bonds.
You got it.
Those hydrogen bonds are back.
They're everywhere.
So they're shaping the protein.
Precisely. And there's more.
Oh.
Those secondary structures fold into even more complex 3D shapes.
Those are the tertiary structures.
You got it.
And this complexity is key to a protein's function.
It is. But this all sounds so complex.
It does.
Do you have a specific example?
You mean of how understanding protein structure is useful?
Exactly.
Well, think about beta-lactam antibiotics.
Like penicillins and cephalosporins.
Yeah. They inhibit bacterial cell wall synthesis.
By targeting penicillin-binding proteins.
So they're like wrenches.
Jamming up the machinery.
Preventing the bacteria from building their walls.
Exactly.
Yeah.
Essentials describe how the beta-lactam ring.
The core of these antibiotics.
Right. It gets hydrolyzed by the bacterial enzyme.
And that leads to its inactivation.
You got it.
So by understanding the enzyme, we can design drugs to disable it.
And kill the bacteria.
It's incredible. So understanding these tiny molecules
Yeah.
is crucial for developing medications.
It is. Now, what about the molecules that carry our genetic code?
DNA and RNA.
Exactly. Those nucleic acids.
Right. They're made up of nucleotides.
Each containing a sugar, a phosphate group, and a nitrogenous base.
Essentials focuses on how amino acids get attached to tRNA molecules.
A crucial step in protein synthesis.
It mentions the aminoacyl AMP intermediate.
And the ester linkage to tRNA.
Yeah, that attachment process ensures the correct amino acid sequence.
Right. It's like a molecular translator.
Making sure the code is read correctly.
Precisely.
The book also touches on chemical sequencing of DNA.
Oh, yeah.
It involves selectively cleaving DNA and labeling the fragments.
To figure out the order of nucleotides.
So we're talking about reading the genome.
Exactly.
Wow. So from sequencing DNA to understanding antibiotics, organic chemistry is everywhere in medicine.
It's fundamental.
This has been an amazing start to our deep dive.
We've covered a lot of ground.
But we've only just scratched the surface.
There's so much more to explore.
We'll be right back with more organic chemistry goodness after a short break.
Sounds good.
Welcome back to our deep dive into organic chemistry. I'm still trying to wrap my head around all these concepts.
It's a lot to take in.
It is. We were talking about tautomers before the break.
Right, those molecules that can shift between different forms.
Essentials of organic chemistry uses those 1,4,3-decarbonyl compounds like ethyl acetoacetate as examples. And they point out how the enol form is so important in those cases.
Yep.
But why? Why does that enol form matter so much?
Well, it's all about reactivity.
Okay.
Remember, the enol form has that alcohol group.
Right.
And that alcohol group can act as a nucleophile.
Meaning?
It's attracted to electron-deficient areas in other molecules.
Ah.
So it can participate in all sorts of reactions to form new bonds.
And it also has that conjugated system.
Right, which adds stability.
So it's like the enol form has this whole toolkit for reactivity.
Exactly. The keto form just doesn't have that.
So by shifting to the enol form,
Yeah.
these molecules can do all sorts of new things.
Exactly. It opens up a whole new world of reactions.
And they can create building blocks for more complex molecules.
They can. And chemists can even control the equilibrium.
Between the keto and enol forms.
Yeah. To fine-tune a molecule's reactivity.
That's incredible. It's like a molecular switch.
I like that analogy.
Turning reactivity on or off.
Precisely.
But March's Advanced Organic Chemistry mentions that the keto form, it's usually favored for most carbonyl compounds.
Right.
So why is that?
Well, it comes down to bond strengths.
Okay.
The carbon-oxygen double bond in the keto form, it's just more stable than the carbon double bond in the enol form.
Ah, so it's an energy game.
It always is.
The keto form is just lower in energy.
Exactly. But those one-cell's 3D dicarbonyls, they get that extra stabilization.
From conjugation and hydrogen bonding.
You got it.
So they can tip the balance towards the enol form.
Right. It's like a molecular tug of war.
I like that. Now, we were talking about pH earlier.
Yeah.
And essentials of organic chemistry used acetic acid as an example.
For those pH calculations.
Right. Why acetic acid?
Well, it's a weak acid.
Which is...
It doesn't completely ionize in solution.
Right.
Plus, it's super common.
Yeah, it's in vinegar.
And it's found in lots of biological systems.
So understanding weak acids is important.
Oh, it's crucial. Because they're everywhere.
And the pH of a solution?
Yeah.
It depends on the acid's concentration too, right?
It does. Diluting an acid increases the pH.
So it becomes less acidic.
Exactly. And concentrating it lowers the pH.
Makes it more acidic.
Right.
It's like adding water to lemonade to make it less tart.
Perfect analogy.
Now, those fused ring systems?
Like decalins and hydrandane.
Yeah, we said the cis isomer of hydrandane is more stable.
Right.
But how does that stability difference actually affect its reactivity?
Well, generally speaking, more stable molecules are less reactive.
Why is that?
They're already in a lower energy state.
Because that's that lol. So they're happy where they are.
You could say that.
So you'd expect the cis isomer of hydrandane to be less reactive.
Then the trans isomer, yeah.
Makes sense. It's like trying to push a ball uphill.
The one at the bottom will be harder to move.
Now, Marches talks about these tormented aromatic systems.
Oh, yeah.
What makes them so tormented?
Well, it's a bit of a dramatic term,
Okay.
but it describes aromatic systems that are forced out of their preferred geometry.
So like a benzene ring that's been bent or twisted?
Exactly. Imagine it fused to other rings that create strain.
Right, like those examples in the book with those really long names.
Yeah, like 7-circuline and 9-phthalate-tribenza, CIG-xanthosine.
I can't even pronounce that one.
Me neither. But the point is, those molecules still exhibit aromaticity.
So they have that special stability.
Right. But the distortion of the ring system introduces instability.
So they're tormented because they're caught between these two forces.
Exactly. They want to be flat and stable.
But the overall structure forces them into a strained conformation.
It's a fascinating concept.
It is. Now let's go back to those antioxidants like vitamin E. We talked about how they interrupt radical chain reactions.
Right.
But can you break down those chain reactions for me?
Sure. It starts with an initiation step.
Where a radical is formed.
Exactly. Often from a weak bond breaking apart.
Yeah.
That radical reacts with a stable molecule.
Like by abstracting a hydrogen atom.
Precisely. So now you have a new radical.
Which can then react with another stable molecule.
And so on and so on.
It's like a domino effect.
It is. And these chain reactions can cause a lot of damage.
Especially in biological systems.
Right. Lipids, proteins, even DNA can be attacked.
And that's where antioxidants come in.
To the rescue.
They act as radical scavengers.
Breaking the chain.
So vitamin E, with its phenolic hydroxyl group, it donates its hydrogen atom to a radical,
stabilizing it
and preventing further damage.
It's a true hero.
Now, speaking of protecting molecules, let's go back to those branched carbohydrates.
Like amylopectin and glycogen.
Yeah, we said branching leads to more compact structures.
Right.
But what other benefits are there?
Well, branching increases the number of non-reducing ends.
And those are?
The sites where glucose units can be added or removed.
Ah, okay.
So having more of them allows for faster mobilization of glucose.
When energy is needed.
Exactly.
It's like having multiple entry and exit points.
For glucose.
Makes it much more efficient.
It does.
Now, what about those different types of glycosidic linkages?
You mean like alpha-1,4 and alpha-1,6?
Yeah. Does the type of linkage really matter that much?
Absolutely. It affects the overall shape and properties.
Can you give me an example?
Sure. Amylose, with its alpha-1 to 4 linkages, it forms a helical structure.
Okay.
Whereas amylopectin, with its alpha-1 to 6 branches, it has the more globular tree-like structure.
So these structural differences, they influence how these starches are digested.
Right, absorbed, yeah.
That's amazing how something as small as a bond orientation
can have such a big impact.
Now, what about cellulose?
Ah, the stuff that makes up plant cell wall.
Right. It's also a polymer of glucose.
It is.
But it has those beta 1 to 4 linkages.
Right, which create long straight chains.
So it's tough.
It is. Makes a great structural material for plants.
But we can't digest it?
Nope. Our enzymes can't break those beta linkages.
It's a good thing for plants, I guess.
It is. Keeps them from being eaten too easily.
Now, those building blocks of proteins?
Amino acids.
What makes each amino acid unique?
Each one has a unique side chain.
Okay.
And that side chain gives it specific properties.
So some are hydrophobic?
Meaning they repel water.
Like oil and water.
Exactly. Alanine and valine are good examples.
They tend to cluster together inside proteins.
Away from the water, yeah.
And then you have hydrophilic amino acids.
The ones that love water.
Like serine and aspartic acid.
Right. They're usually found on the protein surface.
Interacting with the surrounding water.
Exactly.
So it's this interplay between hydrophobic and hydrophilic.
That drives protein folding and stability.
And we can't forget the charged amino acids.
Like lysine and glutamate.
Right. They can form ionic
And
interactions.
those are important for enzyme activity.
Where are charged amino acids in the active site?
Interact with the substrate.
Yeah, it's like a molecular dance floor
I like that.
with different amino acids partnering up.
Based on their charges and affinities.
Now, how does a linear chain of amino acids actually fold into that complex 3D structure?
That's the million-dollar question.
It seems so complicated.
It is. Protein folding is driven by a whole bunch of forces.
Right.
Hydrophobic interactions.
Right.
Hydrogen bonding, electrostatic interactions, van der Waals forces.
So everything we've been talking about is at play here
It all comes together.
to guide the protein into its final shape.
It's pretty remarkable.
It is. Now let's talk about enzymes.
Those amazing biological catalysts.
Right. They're proteins,
They are.
but what makes them such efficient catalysts?
They're incredibly specific.
Meaning?
They only catalyze very particular reactions. They have an active site
Yeah.
that's perfectly shaped to bind to their target molecule.
The substrate.
Right. It's like a lock and key.
So they fit together perfectly.
And then the enzyme helps the reaction go faster.
But how?
It stabilizes the transition state.
The high energy intermediate.
Exactly. And that lowers the activation energy.
Making the reaction happen much faster.
Precisely.
So the enzyme smooths out the path for the reaction.
You could say that.
And this efficiency is a result of evolution.
Billions of years of fine-tuning.
And what about enzyme inhibitors?
Ah, yes.
The molecules that can block enzyme activity.
Right. They can either bind directly to the active site.
Blocking the substrate.
Exactly. Or they can bind to a different site.
And change the enzyme shape.
Or function. Yeah.
So they're like saboteurs.
Interfering with the enzyme's job.
Like those beta-lactam antibiotics. They target those penicillin binding proteins.
Right. By inhibiting those enzymes,
Yeah.
beta-lactams prevent bacteria from building their cell walls.
Leading to bacterial death?
It's a classic example of drug development.
Based on understanding enzyme mechanisms.
Exactly.
Now let's zoom out a bit
Okay.
and talk about metabolic pathways.
Those intricate roadmaps of metabolism.
What are they exactly?
Think of them as a series of interconnected reactions
Okay.
that transform molecules within a cell.
So they're like assembly lines.
Exactly. Molecules are modified step by step, guided by enzymes,
So it's like a bustling factory inside the cell
with different pathways working together.
to break down nutrients
Produce energy.
and synthesize building blocks.
It's all happening at once.
And Essentials gives us a glimpse into the citric acid cycle.
Right. A central hub for energy production.
It oxidizes acetyl-CoA,
And releases energy in the form of ATP.
the cell's energy currency.
Exactly.
But there are other important pathways too, right?
Oh, absolutely.
Like glycolysis.
Which breaks down glucose into pyruvate.
And then gluconeogenesis.
Which makes glucose from non-carbohydrate sources.
These pathways are crucial for keeping us going.
They are.
It's also interconnected.
And beautifully orchestrated.
Now, we've talked about analyzing molecules.
Yeah.
But what about creating new ones?
Organic synthesis.
Right. It's like molecular architecture.
Exactly. Chemists use their knowledge
To build complex molecules?
from simpler starting materials.
It's like assembling molecular Legos.
I love that analogy.
Essentials mention the aldol condensation.
A useful reaction for synthesis.
What other tools do chemists use?
Oh, there's so many. The Grignard reaction.
For forming carbon bonds.
Right. And the Diels-Alder reaction.
To building rings.
Exactly. And then there are reactions for adding functional groups.
Like halogenation, oxidation, reduction.
It's a toolbox of reactions.
So organic synthesis has led to all sorts of advancement.
In medicine, materials science, countless other fields.
It's incredible what chemists can create.
It really is.
This is also fascinating.
It is.
But we're just scratching the surface, right?
Oh, absolutely. There's so much more to explore in organic chemistry.
Well, like, we haven't even talked about stereochemistry.
Or spectroscopy.
Or reaction mechanisms in detail.
It seems like the possibilities are endless.
They really are. There's a whole world of molecular wonders out there.
Well, I'm ready to dive even deeper.
Me too.
Let's continue this exploration in part 3.
Welcome back to our organic chemistry deep dive. We were just discussing enzymes and how they can be so specific.
Oh yeah, those remarkable catalysts.
I'm still thinking about that concept of procurality.
Yes.
Yeah.
Where an enzyme can differentiate between seemingly identical groups.
Like in the example of citrate from essentials.
Right. It has those two digi-CH2-COH groups.
They look identical on paper.
They do.
Wow.
But when citrate binds to the aconitase enzyme, they're actually positioned differently in 3D space.
So the enzyme sees them as different.
Exactly. And that's crucial for the enzyme's function.
Because?
Well, the active site of aconitase, it's shaped in a way that only allows one of those digi-CH2-COH groups to be in the right orientation for the dehydration reaction.
So even though those groups are chemically identical, their position matters.
It makes all the difference.
It's like having two keys that look the same.
But only one actually turns the lock.
Exactly. Procurality really highlights the importance of 3D thinking.
It does.
Now, Marches mentions this thing called kinetic resolution.
Ah, yes. A clever technique.
For separating enantiomers.
Right. It takes advantage of their different reaction rates.
Enantiomers being those mirror images.
Exactly. And they often react at different speeds with chiral molecules.
So if you have a mixture of enantiomers and you react it with a chiral reagent,
one enantiomer might react much faster
than the other.
Exactly. And that's the key to kinetic resolution.
So you can partially separate them.
Right. The faster reacting one gets consumed more quickly.
Leaving behind the slower reacting one.
It's like a race.
Where one runner is much faster.
And you can separate them based on how quickly they finish.
I like that analogy. But this isn't just theoretical, right?
Oh, no.
Yeah.
It has tons of real-world applications.
It's like?
Well, the pharmaceutical industry uses kinetic resolution all the time to produce enantiomerically pure drugs.
Because many drugs are chiral molecules.
Right. And often only one enantiomer has the desired effect.
The other one might be inactive.
Or even harmful.
So they use kinetic resolution to isolate the good enantiomer.
And get rid of the bad one.
To make medication safer and more effective.
Exactly.
Now let's talk about those organometallic compounds from Marches.
Ah, yes. Those fascinating molecules.
What makes them so special?
Well, they have this unique bond.
Between?
A carbon atom and a metal atom.
And that gives them unusual properties.
It does. They often act as powerful nucleophiles or bases.
So they can do things that regular organic molecules can't.
Exactly. They open up new possibilities for reactions.
Like those Grignard reagents we talked about?
Right. With that carbon-magnesium bond.
They're so useful in synthesis.
They are. And Essentials also mentions organometallic compounds.
In what context?
Reactions like the Corey-House synthesis. A way to build larger alkanes from smaller alkyl halides.
So organometallic chemistry has really expanded our synthetic capabilities.
It has. It's a powerful tool.
Now, I also remember Marches talking about these cage complexes.
Oh, yeah?
What are those all about?
They're really interesting. They fall under host-guest chemistry.
Where you have a host molecule
With a cavity or cage-like structure.
that can trap a smaller guest molecule.
Exactly. Marches talks about cyclodextrins.
Right, those cyclic oligosaccharides made of glucose.
They have this cool shape.
Like hollow, truncated cones.
Exactly. And that shape allows them to trap smaller molecules.
And the interaction between host and guest
Yeah.
is driven by weak forces.
Like van der Waals interactions, hydrogen bonding, hydrophobic interactions.
Like a molecular embrace.
I like that.
Well, what's the point of these cage complexes?
I have lots of applications.
Right.
Well, in drug delivery, for instance,
Mm-hmm.
cyclodextrins can encapsulate hydrophobic drugs.
Making them more soluble.
Exactly. Easier for the body to absorb,
So it's like a molecular vehicle.
delivering the drug to its target.
Amazing. And cage complexes are also used in catalysis.
And sensing.
Even material science.
They're incredibly versatile.
It's amazing how these molecular cages can be so useful.
It is.
Well, we've covered a lot of ground in this deep dive.
We have. From simple hydrocarbons to complex biomolecules.
It seems like the possibilities in organic chemistry are endless.
They really are. It's a vast and ever-evolving field.
But it's all based on those fundamental principles.
Right. Bonding, functional groups, stereochemistry, reaction mechanisms.
It's like learning a new language.
The language of molecules.
And by understanding this language,
We could design new molecules, new materials, new technologies.
and ultimately understand life itself.
Exactly. It's all connected.
This deep dive has been an incredible journey.
It has.
I feel like I've gained a whole new appreciation for organic chemistry.
I'm glad to hear that.
It's been a pleasure exploring these concepts with you.
Likewise.
And to all our listeners out there,
Keep learning. Keep questioning.
and keep marveling at the wonders of the molecular world.
Absolutely.
Thank you for joining us on this deep dive into organic chemistry.
It's been a blast.